So the topic of discussion now is the development of the specific and unique receptors that are found on the surface of your adaptive lymphocytes. Remember, we spoke before about the production in the bone marrow of two categories of lymphocytes, innate and adaptive. And within the adaptive branch, we have B cells and T cells. Now, in the generative or primary lymphoid organs, what these cells are going to do is undergo a complex set of rearrangements of gene segments that is going to allow them to create their unique receptor for foreign substance or antigen. And this needs to be done because of the fact that you have a relatively small amount of DNA which encodes the production of different cell receptor molecules, the B cell receptor and the T cell receptor, and yet you have an almost unlimited number of different specificities and shapes of those molecules. So very early on, they realized that there simply could not be the case that this followed the one gene, one protein theorem. So what they then discovered was that during the development of adaptive lymphocytes, they undergo these rearrangements and will take segments of genes and shuffle them in such a way that they create the unique ending pocket that we before referred to as the idiotype. So notice, if we're talking about this happening within the primary lymphoid organs, what segments we have are called variable and diversity and joining. Now, those are simply segments of genes. These are DNA segments, and you don't have to get too anal retentive about this process as long as you understand that what I'm trying to do is generate a diverse number of different specificities so that in a given day in my life, I have the ability to make responses to things that are very wide in scope, and I have not indeed seen them before. So you don't need to know how many there are, but let's just say there are hundreds of variable and diversity and joining. So you can imagine if they're each one slightly different in DNA sequence, they're going to generate a different sequence of amino acids. And remember that what we're trying to create here is the very N-terminal end, the variable domain of the molecule. And remember, both my T cell receptor and my B cell receptor have a variable domain at the N-terminal end of their receptor, so that when we rearrange the amino acids by shuffling the DNA, we're going to make a different shape to the pocket that will then accommodate a foreign substance. Now, while this is going on, it's under the control of a group of genes called RAG genes, and that stands for recombination activating genes. And what will happen is that the random choice of a V and a D and a J will be spliced together in a unique way so that the cell that has completed that splicing now has a prescribed sequence of amino acids that would be in that N-terminal end. In addition to that, there is an enzyme known as terminal deoxyribonucleotide transferase, which is quite a mouthful, but TDT is an enzyme that randomly adds bases in every time there is a splice between a V and a D and a J. Now, for any sensible biochemist, this would sound horrifying because, of course, we invest a tremendous amount of energy into proofreading as we replicate DNA. But what I'm saying here is that this enzyme randomly, without an equivalent molecule to read on the coding strand, is simply randomly adding bases. And so this adds even another layer of random diversity into the formation of the receptor, and indeed is a possibility for causing mutations that may result in a non-functional protein being made. So obviously, I think you should see that the designer of this process wanted you to be able to generate a huge diversity of different molecules and is willing to take the risk that some of the molecules that you would make would result in a non-functional cell, which would then be induced to undergo apoptosis in the primary lymphoid organs. Now, as I said, I don't want you getting too obsessive compulsive about the process here, but in broad strokes, if I were looking at the germline DNA and reading from 5' to 3', I would have in order a group of variable gene segments, a group of diversity gene segments, and a group of joining gene segments. And the first thing that is going to happen is that under the influence of the RAG genes, the cell is going to pick a random D and a random J, and it's going to loop up the DNA intervening between them and cut it out. So that is going to cause an irrevocable choice of that single D and single J, which are now joined together. And then the next process will be that a single V will be chosen out of many, and that V will then be joined to the selected D and J, again with the intervening DNA cut out. So at the pre-messenger RNA level, notice that I am now fixed in what I have for my N-terminal 110 or so amino acids on that variable domain. And because of the fact that each of these steps required excising DNA in between the selected gene segments, it means that I cannot change my mind and go back and select another one. This is an irrevocable stepwise process that is done in the absence of any antigenic stimulation. And the point of the whole process is to create the maximum size of orchestra of different types of molecules so that you have the potential for recognizing a diversity of different invaders. Now, downstream from that, as we'll talk more about later, we have in sequence the constant domains. And the first one after the coding for VDJ, the idiotype, is going to be the constant for mu. So by definition, the first heavy chain I'm going to make is that of IgM. And you should realize that that's an incredibly important clinical fact, because as you'll see when we discuss serology and diagnosis of many infectious diseases, you're going to always realize that regardless of the agent or the timing of the infection, you're always going to make IgM first. After IgM is made, you may receive signaling to cause production of other isotypes, but there is no possibility of ever making anything other than IgM first. So we always use that as our marker of a primary or early infectious process. Now, this was the discussion of what happens with the rearrangement of the heavy chain genes. And remember that the analogy between the B cell receptor and the T cell receptor is quite strong. So a very similar process would be going on in the beta chain of the T cell receptor. And I always remember this by sort of synonymizing heavy and big, and then spelling big with a beta in front. So that will remind you which of the two chains are analogous to one another. So by contrast now, realize that my light chain on my B cell receptor is going to be analogous to the alpha chain on my T cell receptor. So when I look at what goes on here, notice that one key difference is that I have no D gene segments. I have variable and I have joining, but I do not have diversity. So I only need to make one set of choices. I will choose randomly a variable and a joining. I will loop up the intervening DNA and slice it out. And at the point of the rearranged DNA, I will have that variable and joining that I selected irrevocably joined together. The remaining DNA has been excised. And now downstream from that, I have the coding for the kappa constant domain. So a very similar process, but only 2 segments out of which to select. And remember again, I am now simply creating the coding for about 110 amino acids on the N-terminal end of the light chain or the alpha chain. Obviously, when I put the heavy and the light together, those 2 variable domains create the 3-dimensional shape of the idiotype. So I have created the antigenic specificity of this molecule for the remaining life of the cell. And so it's important to realize that this is not something that can be undone over time. If I have created a cell that is an anti-slide advancer cell because of the shape of its idiotype, I cannot stimulate that cell and cause it to produce another type of antibody that recognizes some other sort of molecule. That cell, for eternity of its life, can only create a receptor site that would be specific for the antigen for which it has 3-dimensional complementarity. So from here, we need to realize that as we talked about the addition of the random nucleotides by terminal deoxyribonucleotide transferase, if I add 1 nucleotide at each splice point, realize in that case I have created a change in the reading frame. If I have added 2, similarly I've changed the reading frame. If I add 3 bases, then I've added another amino acid. Now, one of the things that's different between T cells and B cells is that in the case of a B cell heavy chain, TDT is active. That is true also for the beta chain of the T cell receptor. But in the case of the light chain rearrangement with the B cell receptor, there is no activity of TDT. For the T cell receptor, there is activity of TDT. So notice that one would argue that the designer of the system actually wanted T cells to have more possibility for diverse shapes of receptors, probably again because, as I've said before, the T cell is going to be the general of your immunologic army, and his or her ability to see foreign substances needs to be the most astute of any of the cells in your system. Now, because of the fact that this process is likely to be fraught with possibilities of errors, realize that if I make an abnormally shaped or a truncated protein or an abnormal protein of any kind that cannot take its position on the cell membrane, cannot bind to things that it needs to bind to, then that cell that has done that process will be induced to undergo apoptosis. And this is what we refer to as allelic exclusion. The reason being that the cell has multiple possibilities of doing this right. If the cell randomly rearranges the paternal chromosome first and makes a goof and cannot make a productive protein, then that chromosome would be shut off, but then the complementary one of that pair would be used to try again. Regardless, whichever chromosome succeeds in creating a functional product, that chromosome will be left turned on. The other one of the pair will be turned off or excluded. And what this means is that in a given cell, again, only one type of B cell receptor or T cell receptor is going to be expressed. I don't want schizophrenic lymphocytes who see one thing with one type of receptor and another thing with another type of receptor. Now, one final point here, realize that my heavy chain genes are all on one chromosome, and the light chain genes are on two separate chromosomes. So with each of those choices, I have a paternal and a maternal, and therefore two options to get the process right. Any cell that is incapable of doing this properly and makes a non-functional receptor will be removed from the population in the bone marrow by apoptosis and will not be allowed to leave that location. Now, the final step in this process, as I alluded to before, is the addition of the constant domains of the receptor. So when we're talking about B lymphocytes, we're talking about those constant domains that will dictate the isotype of the receptor. And in the case of the T cell, we're talking about the alpha and beta constant domains for those types of chains. So notice if we look specifically at the B cell, which is a more complex genetic creature, notice that my idiotype choice here at the beginning of 1V and 1D and 1J is then followed immediately downstream in sequence by all of the constant domains that the B lymphocyte will ever be able to make in its lifespan. As I said before, the first is going to be the one that calls for production of IgM. And as that is created at the RNA level by alternative RNA splicing, the next reading through the idiotype domains will be joined to the constant domains for delta, which will give you an IgD molecule. Now you'll notice that we don't talk a lot about IgD because, believe it or not, we still do not know what IgD does. But suffice it to say that when we have a mature, naive lymphocyte, meaning a fully functional B cell coming out of the bone marrow, but has not done anything responsible yet, that mature naive B cell has equivalent amounts of IgM and IgD on its surface. And that's the only time in the B cell life that it actually carries two isotypes of antibody at the same time. Now, all of these constant domains downstream in the DNA are under the control of help from T cells. So I will not make anything but IgM and IgD until I receive signaling from a helper T cell that would then activate one of these regions and cause the idiotype to now be joined to another constant domain. Now, the process of constant domain addition is quite similar to what we saw before. I take the idiotype, I loop up the DNA, and I excise the intervening segment. So notice that my B lymphocyte will go unidirectionally down this sequence. It does not have to do it step by step by step, and that is totally dependent on T cell help. So whatever the T cell tells the B cell to switch to by signaling, that B lymphocyte will change to a new isotype of antibody. But realize I cannot go backward in this process to making IgM and IgD again, because at each step I'm cutting out the constant domains that I'm no longer using, so it's a one-way street developmentally for the B cell to create different isotypes. Now, you don't need to notice the sequence as far as what isotypes and subisotypes come next. That will be the subject of a different discussion later on. But for now, simply realize that M and D are what the cell will emerge from the bone marrow possessing, and when T cell help arises, then I will be able to make the other isotypes of antibody. And as I said to you before, I've got 4 subisotypes of IgG, so that tends to be the most common isotype made in the body. I have 2 of IgA, so that would be next most likely. And notice way downstream here at the end of the DNA is the coding for epsilon, which, if you recall from previous discussions, is the molecule that's involved in both type 1 hypersensitivities and in responses to parasites. So that one is pretty much left to the very end of the germline DNA because it is an immunoglobulin that tends to have harmless side effects. And one wouldn't want to make IgE unless there was a very strong reason to do so. So now, I know some of this sometimes makes you feel as though there's a tremendous amount of molecular detail, and yet very little obvious pertinence to how good a physician you will actually be. But I wanted again to introduce my Make Me Care quotient here and talk with you about why you should at least understand the broad strokes of what's going on here. I'm trying to generate a maximum number of different receptors on my lymphocytes, which are then going to serve to protect me throughout my life from things that I have not necessarily seen immunologically as yet. So think about for a moment, if you had a scenario where a patient did not have the ability to create RAG gene products, can you imagine what that would do to the immune response? I'm going to suggest now that you pause the video and think about this and see if you can't explain to me what would happen if I had that inability in a child. Well, if the child can't do recombination at the VDJ level, realize that a cell that does not have a functional receptor has no value to your body. So this means that every single cell being created on the lymphoid and adaptive lineage side would end up being induced to undergo apoptosis in the bone marrow. So this quite clearly, I think you can see, would result in a severe combined immunodeficiency. This is the classic bubble baby that can only be dealt with by bone marrow transplantation to reconstitute the ability of cells to do this. So this is a very dire immunological problem. My second question to you concerns a neonatal screening test that we haven't talked about yet, but it again is a screening test for severe combined immunodeficiencies. And there are actually many of them, but this particular blood test on the child takes a drop of dried blood and then tests it for detection of T cell receptor excision circles, or Tregs. Now, I want you to think about that word, T cell receptor excision circles. Think about what we've talked about in our discussion here, and then come up with a logic for why that would be a useful assay to measure and predict whether a child has normal thymic cell function, T cell precursor function. I would pause the video now, give yourself a few minutes to work through the process of why this would matter, and I will continue when you click the non-pause button again to say to you what this assay is measuring is actual episomal DNA that has been excised out of the germline DNA in the process of VDJ recombination. And these episomes actually persist in the bloodstream for as long as a child and adolescent is producing normal numbers of T lymphocytes. So what you're measuring is the actual function of VDJ recombination happening in the thymus. And so if I were to ask you, would I want high numbers of excision circles to be detected or low numbers? The healthy individual would have a standard number that would be quite high for the number of Tregs detected in a blood sample, whereas a person with an immunodeficiency, such as a childhood SCID, would be one that would have very small numbers of these. So there are two places that we will be talking about this process again as we go forward and talk about both diagnosis of genetic disease and indeed the Omenn syndrome, which will be the subject of a clinical case that I will do with you.